Hi, my name is Oliver Yu at the Danforth Plant Science Center. Today, we are going to show you how to generate hairy root composite plants by Agrobacterium rhizogenes. This technique was brought to Danforth Center by Dr. Chris Taylor, currently at Ohio State University. Several of my former group members improved these techniques over the years. And today, my current group member, Drs. Deng and Mao, put together this video to show how hairy root composite plants is produced. Agrobacterium rhizogenes are very similar to Agrobacterium tumefaciens in that they introduce foreign DNA into plants, but the rhizogenes can induce hairy root formation on wild-type tissues, forming composite plants, as this one. So on these composite plants, some of the roots are transgenic while the shoots are still wild type. Therefore, this technology will not produce transgenic seeds. However, there are three major advantages of hairy root composite plants that make this technology very useful. First, many plant species, especially dicot species, can be transformed by Agrobacterium rhizogenes. Second, we do not need to establish tissue culture prior to transformation. We can take many parts of plants, such as seedlings, shoots, and cotyledons, to generate composite plants directly. And finally, we can get transgenic root tissue in a matter of weeks. For soybean and Medicago truncatula, we can get transgenic roots in as short as 3 weeks, faster than Arabidopsis transformation. Overall, the hairy root composite plants technology is very useful tool to study gene functions and root-related phenotypes.
Hello, my name is Ying Deng.
Hello, my name is Guo Hongmao.
There is my cameraman, Bill. Today we will introduce hairy root composite plant protocol.
Yeah, we use this protocol in our several projects. Are you ready? We start it. Yeah.
For our demonstration, we will use Medicago truncatula as an example. These are Medicago mature pods. Put the pods on a grinding box and then grind the pods with a long, firm motion. Separate the seeds from the seed pods by gently shaking. Place seeds in a 50 milliliter centrifuge tube, then add 5 milliliters of concentrated sulfuric acid very carefully. Scarring takes about 10 minutes in acid. Vortex the tube 2 to 3 times during the acid treatment. The seed coat will change to a yellow color. Remove sulfuric acid from the tube and rinse the seeds at least 5 times with sterile water. Soak the seeds in deionized water at room temperature for 6 hours, and then keep them at 4 degrees centigrade for 2 days to synchronize germination. Spread 20 to 30 seeds on moist filter paper in the Petri dish. After 5 to 6 days, transfer the seedlings to soil and then grow them in the greenhouse for 4 weeks. For composite plant transformation, we normally use the Agrobacterium rhizogenes K599 strain. The shuttle vector, or binary vector, can be transformed into the K599 following the standard Agrobacterium tumefacient transformation protocol. Inoculate the Agrobacterium. Grow them at 30 degrees centigrade for 16 to 24 hours. After the growth period, collect the cells by placing the culture solution in a 50 milliliter centrifuge tube. Run it in the centrifuge at 5000 G for 30 minutes. Resuspend the pellet with nitrogen-free nutrient solution. Make sure to dilute the agrobacterium cells to 0.3 at OD 600. This concentration is important for maximum transformation. We use rock wool as a supporting matrix. Using this material is important for high-frequency transformation. Cut the rock wool into the right size plug. Poke a hole at the top about 1.5 to 2 centimeters in diameter using a pipette tip. Add 5 milliliters of resuspended Agrobacterium rhizogenes solution into each plug. Select a stem section with 2 to 3 auxiliary buds and make a slanting cut. Insert the plant sections into the hole in the rock wool. Place the cut plants into a growth chamber or lab bench for 3 weeks. Add 5 milliliters of nutrient solution into each plug every 10 days. After 3 weeks, the roots are regenerated from the cut side. Soak the plant in water. Remove the softened rock wool carefully and do not damage the roots. Be sure to remove all of the rock wool. You can transfer the hairy root composite plants to sand or perlite. For symbiosis experiments, grow the rhizobium in a yeast extract mannitol medium for 7 days at 30 degrees Celsius. Collect the rhizobia cells. Resuspend the pellet with the nitrogen-free nutrient solution. Dilute the solution to 0.08 at OD 600. Add 10 milliliters of rhizobia solution to each composite plant. Culture the plant for an additional 2 to 3 weeks to allow nodules to form. Remove sand from the composite plants by uprooting and washing them. Use green fluorescent protein expression to check the transgenic root. You will use a fluorescent microscope to do this. All our hairy root constructs have a constitutive GFP cassette for screening transgenic lines. This picture shows the GFP transgenic hairy root composite plants observed in the fluorescent microscope. The transgenic roots with the brighter green fluorescence are easy to distinguish from the non-transgenic roots. Nodules are formed in the hairy root of the composite plants.
Here is the complete protocol. Once again, generating hairy root composite plants is a quick and easy method to obtain large quantities of transgenic tissue, especially for dicot species. Although this method cannot give you transgenic seeds, it can produce transgenic material in a few weeks. It's especially suitable for plants that have difficulty in establishing tissue culture and transformation. Over the years, we have used this technology to study gene functions, promoter functions, microRNAs, root and lateral root developments, defense and abiotic stress response, nodulation and other symbiotic process. We studied hormone response, metabolic profiling, gene profiling, and proteomic analysis using the hairy root composite plants. We hope this video will help you adapt this system to your favorite species. You are welcome to contact us for questions related to this technology, and good luck.